So, in this segment we will talk about the subcortical structures. So, we said last time that the cortex is the sheet of neurons that covers the entire cerebral surface. Subcortical structures are basically those that lie under the cortex or the subcortex. So, here some of the main subcortical structures which we will be talking about are the thalamus, hypothalamus, basal ganglia. It is not one structure, but it is a whole circuit where several modules together are called the basal ganglia. Then there is hippocampus or the hippocampal complex, which again consists of lots of neural clusters or the hippocampal fields. Then we will also talk about cerebellum. Cerebellum is not exactly a subcortical structure, because it is not located under the cortex, it is outside the brain, located ventral to the occipital lobe. So, but we will also it is not cortex. So, we will talk about this also in the same category. So, in this figure you can see thalamus. It is this egg shaped pink structure. So, thalamus is a major important gateway to the cortex. Most sensory information that goes to the cortex goes through the thalamus. So, sensory information and also motor integration occurs through thalamus. So, for example, the motor output from the motor cortex does not go through thalamus, but the motor cortex is influenced in its motor in its movement decision making by other structures like basal ganglia or cerebellum, and these influences reach the motor cortex via the thalamus. So, both on the motor side and sensory side, a lot of information has to go through the thalamus. And thalamus consists of 50 nuclei. This nucleus here is basically a cluster of neurons. It is a compartment within the thalamus. There are 50 of them approximately, and here there are two kinds of nuclei. There are specific nuclei which make point to point connections to the cortex, and they also receive feedback connections, extensive feedback connections from the cortex. Then there are these non-specific nuclei which send diffuse projections to the cortex. So, let us talk about both these kinds of nuclei. So, this picture actually shows some of the connections to the cortex from the sensory organs, and and then the connections from the thalamus to the cortex. So, we talked about lateral geniculate nucleus, right. So, visual information from the eyes comes to lateral geniculate nucleus, and then from there it goes to the primary visual cortex. So, similarly, medial geniculate nucleus comes from the information to this structure comes from the ears, right, and then from there it goes to the primary auditory cortex, and so on and so forth. These are some of the examples of point to point connections. So, you can think of the thalamus as some kind of a hospital reception desk, where patients come into the hospital and they do not directly go to different divisions or different departments of the hospital. They go to the receptionist desk, and then the receptionist directs them to which division they are supposed to go to. So, let us talk about point to point connections. So, to take as an example, consider retina on which the input image falls, and information from retina goes to LGN, and then from there it goes to V1. So, what we mean by point to point connections is, so if you place a small dot of light on the retina, right, that produces a very specific image on the LGN, that is a small local cluster of neurons get activated in response to this dot, and then when the LGN projects to V1, then another local cluster of neurons in V1 get activated. So, what that means is every point on the retina gets mapped onto a very specific point on the LGN, which in turn gets mapped onto a specific point on V1. So, there is a point to point mapping between retina and V1, and which is mediated through LGN. So, this is what we mean by point to point connections from thalamic nuclei. So, as you can see in this cartoon picture, a sensory input comes to both the thalami. There are two thalamuses or thalami plural, right, on either side in either hemisphere, and information from sensory sources comes to the thalamus, and then it is routed to various cortical areas. Now, one logical question we can ask at this point is, why do we need the thalamus? Why can't the sensory fibers directly go to the corresponding cortical area, right? Why this stopover? Because in fact, some of the older textbooks on neuroscience simply describe the thalamic function as if it is a relay nucleus. That means, it just simply takes the input and passes it on to a target structure, but why do you need this intermediary nucleus? So, let us try to argue the role for a structure like thalamus by looking at this picture. So, when you look at this picture, you can see that there are 5 horses in it, and each horse is being driven by a horseman, right, and they are like moving at a high velocity, and as you can see from their gait and from the inclination and so on. and you can also notice the colors of the jackets worn by the horsemen, and then the colors of the caps, right, and the colors of the horses. There are black ones, the brown ones, and so on and so forth. So, you look at this picture, obviously you do not take in the entire picture as a whole, right. You shift your attention from point to point on this picture, and whenever your eyes fixate on a given point, they gather information about the part of the image in the neighborhood of the point where you fixated, right. Because if you want to process the entire image as a whole, that requires a lot of computational resources in the brain. So, for example, if you are looking at this image, it is processed by your visual system, right. Your color is processed in say V4, a part of the visual system, and the fact that the horses are moving, suppose you are looking at the real video of the horses, you can see that horses are moving, and that is recognized by motion areas like MT and MST. And the fact that there are people in this picture and there are horses, right, that is recognized by some of these object recognition areas of the inferotemporal area, right. And then in the lower areas you simply look at patches of color and local lines or edges and things and primitive information like this. So, to process to apply all this machinery, all this visual processing machinery on every part of the image simultaneously is expensive and not economical. So, brain figured out a way of doing it piecemeal. You apply the entire machinery of visual processing on one part of the image at a time and complete the processing, and once you are done there, you shift your focus or attention to another part, right. So, you are basically you apply some kind of a spotlight, right, and a spotlight basically highlights a part of the stage, right, which is important at that moment, and once it is done, the spotlight moves over to another part of the stage which is of interest at that moment. There are theories which propose that thalamus plays the role of this kind of a spotlight, an attentional spotlight. So, although the wiring is fixed, there is some kind of a machinery that goes on inside the thalamus, right. This machinery essentially does what you are seeing in this picture. So, in this picture you are highlighting a part of the image, and whereas the surrounding area is blurred. So, you do not have much information coming from surrounding area. You are trying to access as much information as possible from the central area, and by applying some kind of a spotlight in the center. The thalamus applies this kind of a spotlight, right. And now let us come to diffuse projections of thalamus. So, like I said, there are these point to point connections, and from some of the nuclei, thalamus nuclei like LGN and MGN. There are other nuclei like, for example, the internal medullary lamina and the interlaminar nuclei, right here, which can be seen here, and these project to extensive parts of cortex. So, like you can see in this cartoon picture here. Okay, so, what is the purpose of these kinds of nuclei? Right. One example that you can give, there are many nuclei with very diverse functions, but one example you can give is the action of another structure called the reticular activating system, or RAS. The RAS controls your arousal and wakefulness. Some of the systems we have seen before are specific systems, for example, sensory for processing sensory and motor function, whereas the RAS simply controls the overall global state of the brain, because if you want to process sensory information or become aware of sensory information or produce movement, the brain should first be conscious. It needs to be in a waking state, right. In sleep you cannot move much. I mean, you can some people have suffer from a condition called somnambulism, where even in sleep they can walk around, but I mean that is an abnormal condition. But in normal condition, your your antegrav muscles of your body and the legs are deactivated in sleep state. So, you do not get to move much during your sleep other than rolling around. So, brain undergoes a global change in state between waking and sleep, and this change in state is triggered by the controlled by reticular activating system. And since it is a global change, right, the RAS actually projects to the thalamus, and so like you see in this picture, the RAS is located here. It projects to thalamus, right, and through the diffuse projecting nuclei of the thalamus, they activate and access large parts of the cortex. So, here there are again two kinds of connections from RAS to RAS. So, there are the upward connections from RAS to thalamus, and from thalamus up and onwards to the cortex. Then the RAS also sends downward connections to spinal cord and onward to muscles, which control the anti-gravity muscles. These are the muscles which keep us erect, or in a standing position, working against the gravity. Therefore, they are called anti-gravity muscles. So, these downward signals also go to control the anti-gravity muscles. So, in sleep state the anti-gravity muscles are deactivated. So, you do not get to move, you won't be able to stand, whereas in waking state anti-gravity muscles are activated or enabled, so that you can stand erect. So, these signals from RAS control and through thalamus, which through the diffuse projection systems of the thalamus, they control the cortex and change it between the waking and sleep states. The RAS in turn is also can be activated by the peripheral sensory signals, especially pain signals, and that kind of input, this feature you can even verify in your personal experience. Suppose you are very sleepy, right, and you can kind of counter that by simply pinching yourself or rubbing your eyes and things like that. By giving slightly painful stimuli, you can act on your RAS, and RAS in turn sends signal to the cortex, increasing the level of arousal in the cortex. So, therefore, RAS area RAS also receives feedback from the cortex. So, when you pinch yourself, this signal goes to the somatosensory cortex, and then that sends feedback to the RAS, and that in turn changes the arousal. So, another structure which is located ventral to the thalamus is the hypothalamus. Now, just as the thalamus can be considered as a junction point between the world and the brain, you can think of hypothalamus as junction point between the brain and the body, right, because brain is not just receiving information from the world and acting upon the world, it is also simultaneously receiving information from the body and in turn controlling various bodily processes. So, a lot of that control from the brain to the body runs through hypothalamus. So, hypothalamus has extensive connections to the thalamus and to the midbrain and other cortical areas, which receive input from the autonomic nervous system. This is a part of the nervous system which is responsible for controlling the bodily organs, and it is also involved in hormonal secretion through pituitary gland. And I think you can see this picture here. Pituitary gland is not shown here, it is somewhere in front of here in hypothalamus. So, this gland is the master gland. So, the gland system of the body is called the endocrine system, and pituitary gland is the master gland which controls the other glands. So, the hypothalamus controls the pituitary gland. It also controls other bodily parameters like body temperature, emotions, hunger, thirst activities, and circadian rhythms. So, circadian rhythms are intrinsic bodily rhythms which are roughly 24 hours long. So, we know that a lot of the process that you see in the world are driven by the day and night cycle, right, that is driven by the external movements of the sun and all that. But there are internal processes also which are roughly 24 hours long. So, that is why they are called circadian rhythms. There is a separate brain structure called the suprachiasmatic nucleus, which controls the internal bodily circadian rhythms, and so the suprachiasmatic nucleus also talks to hypothalamus and also the RAS to control the sleep wake cycles. So, then there is a structure called basal ganglia, which is also a subcortical structure. It is actually a circuit of several nuclei. You can see some of them here in this coronal section. So, the globus pallidus and the putamen, the light blue region, then the substantia nigra, then there are two of them, there are and the subthalamic nucleus here, the green structure, right, and all these structures together are called basal ganglia. And so, this circuit, right, is involved in lots of functions. In fact, they are involved in pretty much all the four major functions of the brain, that is motor, cognitive, affective or emotional, and also autonomic, or which is related to the functions of the autonomic nervous system. In motor, again they control a variety of motor functions, like for example, movements of the hand or movements of the legs or gait, movements of the eyes or saccades, and so on. So, in cognitive they control working memory or action selection. So, if you have multiple actions among which you have to choose the best action, right, how do you choose that? Based on which action is going to be most rewarding. Okay. So, this function is strongly controlled by basal ganglia, and then language also. Then affective, it controls the reward, loss and risk. In autonomic, it controls gastrointestinal processes and cardiovascular activity. So, one of the diseases which is associated with function of basal ganglia is the Parkinson's disease. It is a progressive neurodegenerative disorder. It is characterized by loss of cells in this one of the basal ganglia structures called the substantia nigra pars compacta or SNC. And there is no cure for the disease, there is only management. You can slow down the disease by use of drugs or brain stimulation and other methods, but to date there is no way of stopping this loss of cells in this part of the brain. And loss of cells in this tiny part of the brain can have very substantial or dramatic effects on symptoms. So, there are 4 major symptoms called the cardinal symptoms of Parkinson's. They are tremor, they are this shaking of hands and other extremities. There is rigidity of the joints, then postural abnormalities. So, very often you see PD patients walking with a little stoop, that is a postural abnormality. And bradykinesia refers to slow movement, slowness of movement. So, they move slowly. In fact, this slowness in movement is also sometimes extended to slowness in thought, right. They think slowly, they react slowly. So, this which is called bradyphrenia. So, brady is slow and phrenia is mind. So, like I was mentioning before, motor symptoms in PD can be linked to movements of reaching, movements of handwriting. In fact, PD patients have this kind of a peculiar handwriting where they write very small letters. It is called micrographia. They also have problems with eye movements and problems in speech and posture and so on. So, within motor, right, pretty much all the forms of motor output or motor modalities can in principle be affected in this disease. In a given patient, you may not see all the symptoms, but in principle all these symptoms can be seen in PD patients just because of loss of cells in this tiny little part of basal ganglia, the SNC. So, an important chemical which plays a lot of coordinating role in the process of basal ganglia is dopamine. It is called the brain's pleasure chemical. It is released by brain stimulation. For example, there are these pleasure centers in the brain, right, which when stimulated electrically by placing an electrode can give a sense of pleasure. The person feels happy if if you can somehow place a wire electrode in that part of the brain and pass current in that part of the brain. So, some of these earlier studies were were done in the 60s and on experimental animals. In some experiments, the animals were hooked up by an electrode like this, and there was a setup by which the animal can just press a lever and which will activate this electrode, and the electrode passes current in the pleasure center. So, the animal kept on choosing this kept on pressing the lever and activating its pleasure centers, and at the cost of foregoing food. So, the animal literally starved itself, right. It just simulated itself through this electrode, but it did not take any food. So, it is a kind of addictive process which is triggered by stimulation of these pleasure centers. Now, when these pleasure centers are stimulated, dopamine is also released. In addition, if you take psychomotor stimulants, drugs, opiates, all these addictive substances also cause release of dopamine. And even food can be addictive. Food is also a pleasurable kind of stimulus. So, therefore, food also causes release of dopamine. And contrarily, application of dopamine antagonists, so that is substances which counter the effect of dopamine, right, attenuate or suppress the pleasurable experience of the above kind of pleasurable stimuli. Therefore, it is thought that dopamine serves as a reward signal. It represents reward, right, in the brain, and there is a lot of evidence to support that. So, the fact that dopamine represents reward, right, has been linked to a certain form of learning called reinforcement learning. So, which has been observed in animal psychology for a long time. So, in reinforcement learning, basically the animal or the agent, right, tries to learn the correct response to a given stimulus by taking the reward or punishment feedback from the environment. So, to take an example, let us say there is an animal which looks at a little say a yellow thing and thinks it is some kind of food item and grabs it and eats it. It turns out that it is a good food item, very nutritive and all that, and tasty. So, next time when the animal sees the same stimulus, same object, it will exhibit the same response and eats it. So, that is what you see in this upper block diagram. So, you have the agent or the animal which looks at the stimulus, in this case the yellow object, and produces a response, and that gave a reward to the animal, because which is probably represented in the form of dopamine signals, and because of that reward the stimulus response connection is strengthened further. right, and next time around for the same stimulus the animal produces the same response. It learns that relationship. Whereas, imagine another case where the animal saw some, I don't know, brown or a red object and grabs it and it gets bitten, right, or stung by that say bug, right, and next time around when the animal sees the same object, it stays away from it or avoids it. So, in this case, for a given stimulus, when the animal produces a response which has led to a punishment or negative reward or negative reinforcement, and therefore, next time around the animal does not produce a response, the same response, and in this case the connection between the stimulus and response is attenuated or suppressed. So, this kind of learning is called reinforcement learning, and there is a lot of evidence that the basal ganglia system exactly is involved in implementing this kind of reinforcement learning. So, if you look at the anatomical position of basal ganglia, it basically takes input from the sensory and motor cortices, and so if you apply the previous, if you refer to the previous slide where there is an agent which takes a stimulus and produces a response, right, within the brain you can imagine that the stimulus information goes to the sensory cortex, right, and then the sensory areas project to motor areas, and the motor areas produce this commands necessary for movement. So, this is like the main pathway, and the information from sensory and motor cortices is also projected to basal ganglia, and basal ganglia also receives information from substantia nigra, right, which is where there are dopamine cells and dopamine is released here. So, when you have a pleasurable experience, dopamine is released from so substantia nigra neurons, and also the other dopamine clusters also, so there also dopamine is released. So, dopamine from substantia nigra goes to basal ganglia. So, you can see that just like in this in a schematic where this box, right, has access to the stimulus information, response information and the reward information. Basal ganglia has access here to the stimulus and response, that is motor and sensory areas coming from the cortex, and it also has access to the reward information which is coming from the SNC. You can combine these three forms of information and then send its output back to the motor cortex and recommending it to strengthen the connection so that it produces the same response the next time around, or recommending it to attenuate the connection so that asking it not to produce the same response next time around when it sees the same stimulus. Okay, so, that is what basal ganglia does. So, when we talk about basal ganglia, we should mention that there are two kinds of memory systems in the brain. So, first is called the declarative memory, which is referred to the knowledge of what, right. Here it is called declarative because this is a kind of knowledge which you can express, like for example, what is the capital of India, or what color is a typical leaf, right, and so on. So, this is a kind of, these are factual information, right, and the other kind of information is procedural memory. This is here this is described as knowledge of how, or this is refers to the memory of motor skills. So, for example, riding a bike, that is something you cannot declare, you just know it, it is in your motor system, you can do it, but it is hard to define it precisely. So, so the basal ganglia is involved in the memory of motor skills or habits, which is a procedural memory, whereas for the other kind of memory, the declarative memory, there is a different structure which is involved, and that is called hippocampus. Okay, and within declarative memory there are two subcategories which refer to the memory of events, and that is episodic memory and or memory of facts, right, which is called semantic or word memory, or refers to the memory of names of things and places and things like that. So, hippocampus is located inside the temporal lobe, as you can see in this figure. So, this long structure is the hippocampus. Its function, there are two important functions. It does memory consolidation, that is when you receive information from outside through sensory apparatus and through the sensory systems, this information is thought to be stored for a short time in the hippocampus, and then from there it is thought to be passed on to long term stores in the cortex. So, therefore, it will plays a major role in memory consolidation. In addition, it also plays a role in spatial navigation, that is how do we find our way around in this three dimensional world and navigate the three dimensional space. So, let us look at that. First of all, let us look at the anatomical position of hippocampus. So, hippocampus receives information from widespread cortical sources. So, here you can see that it receives information from parietal areas, the prefrontal areas, and the temporal areas. I mean, that is pretty much the whole of the cortex. And these projections go to two smaller cortical areas called the parahippocampal gyrus and perirenal cortex, and from there these two areas further project to one cortical area called the entorhinal cortex. So, these are located below the temporal lobe and they are not visible in this view. And so, this entorhinal cortex is the gateway to the hippocampal complex. So, from here information goes to the dentate gyrus here, and then a field called CA3, another field called CA1 and subiculum, and comes back to the entorhinal cortex, and from here it is projected back via the same pathways back to the cortex. So, in a simple schematic, this anatomical relationship can be represented like this. So, you have cortex which projects to hippocampus, and then hippocampus projects back to the cortex. So, you can see that there is a huge reduction right in the size of the number of the number of dimensions involved in this representation. So, hippocampus is also thought to do some kind of a data compression right when it receives inputs from the cortex. Let us present a simple schematic right by which we can explain what kind of a compression the hippocampus does and what exactly does it represent right about the sensory state in the sensory cortices. Let us say you are holding a cricket ball right in your hand, and when you are doing this right in your brain and sensory areas of the brain, many areas are activated. So, you see this large patch of red color right, and then in response to that some of the color neurons of your V4 area are likely to be activated. Then you see that the ball is round right, circular in this view, therefore, you see some of the neurons which represent shape right in so in both in V1 and V2 and also higher up in IT area right, these neurons will get activated. Then since you are holding it, you can touch it and feel it. So, you can also recognize that it is a hard object. So, hardness is also represented in some of the neurons of your somatosensory either primary or secondary somatosensory areas. Now, all these areas are representing the same object, and all these areas through higher cortical areas, through higher sensory association areas project to hippocampus after a few stages. So, in hippocampus there are neurons which respond to this whole combination of sensory inputs, this whole sensory pattern right, and then probably there are neurons which represent this, whose firing activity represents this cricket ball. So, so if you see this color and this shape for example right, that activates this cricket ball neurons so to speak, and once the cricket ball neurons are activated, and then they send feedback to again to the cortex, and then that will activate the other parts of the cortex also. For example, even though you are not holding the ball, if you are just looking at the ball right, the visual features of the ball and activate the cricket ball neurons of the hippocampus, and these neurons when they activate the cortical areas back, they might activate the somatosensory areas also. So, even when you are looking at the ball, you have a kind of a sensation or a feeling or a memory that the ball is a hard object. So, a lot of data about hippocampus has come right from the earliest studies by this patient called HM right, these are the initials of his name, and this HM suffered from severe epilepsy, and so in epilepsy is a kind of a disorder where there is abnormal electrical activity that spreads from point to point in the brain, and usually this activity starts from one point and spreads to typically the whole of the brain, whole of the cortex. And starting point, this trigger point is very often located in the temporal lobe. So, these points are called the epileptic foci. So, a surgical treatment for this kind of a condition is identification of the focus and and surgically lesioning the surrounding area. In some cases, in the earliest surgeries that were performed as a treatment for this condition were somewhat crude, and the surgeons would remove the entire temporal lobe. So, a similar surgery was performed on HM, and along with temporal lobe, his hippocampus and amygdala were also removed. Right. So, because of this, initially the family members and the surgeons did not find anything wrong with the patient, but very soon they noticed that he had very serious memory problems. So, for example, he could not recall memories from events that occurred after the surgery, and that is called retrograde amnesia. He also could not memorize new things, so that is called anterograde amnesia. he could only memorize things for a short window after the event has occurred, like for about 10-15 minutes, but after that the memory is gone. So, for example, if the doctor came to meet him right in the hospital ward, the patient would talk to him, introduce himself and so on, and then if the doctor came back again in the afternoon or maybe the following day, it was like a new experience. The patient didn't have any memory of meeting the doctor on the previous day. So, that is retrograde amnesia, and then the inability to form new memories is called anterograde amnesia. Right. So, that is something about the memory consolidation functions of hippocampus. I just mentioned that hippocampus is also involved in spatial navigation. That is, how do you find your way around, right, in a city or in the spatial three-dimensional spatial world. A lot of this work was also done on rats, because rats are known for their excellent spatial navigation capabilities. For example, you can leave a rat in a maze like this, what you are seeing on this slide, and then after some experience, after some running around in the maze, the rat very often finds a way out. In this case, the motivation for the rat is a piece of cheese that is located outside the maze, and it can find its way around and find the cheese. Okay, so the spatial navigation properties of hippocampus became quite popular in the recent times because of the Nobel Prize that was given in 2014 to work done in this area. So, this popular article on that about that Nobel Prize event talks about hippocampus as an inner GPS. It is called the GPS of the brain, which helps the brain to navigate in space. So, this was given to 3 people, and one of them is John O'Keefe, who has worked on rats and found out that so within the hippocampus. So, in 1971 he has found that within the hippocampus there are cells which respond whenever the animal is at certain location or certain place in the maze. So, in this case you can see this zigzag paths. These are the paths of a rat moving around in a maze, and whenever the rat comes to certain location, the place cells fire. That means, that cell represents that location in the maze. Similarly, later on in 2005, the couple Mary Britt Moser and Edward Moser discovered new kinds of cells. So, these cells also fire in whenever the animal is at some location, but they do not fire only at one location, they fire at multiple locations. So, same neuron fires when the animal is at any of these multiple locations. And what is very intriguing about these cells is all these areas form a very nice periodic spatial pattern, and very often these patterns are hexagonal grids. So, therefore, these cells are called grid cells. So, the grid cells are very intriguing. It is really quite challenging to understand how a cell can respond to such patterns of such great spatial symmetry. So, these are called spatial cells, which respond to different parts of different aspects of the surrounding space, and so that they give some clues to how hippocampus represent the surrounding space and how it enables spatial navigation. Next, let us come to cerebellum. Cerebellum is the structure which is seen ventral to the occipital lobe. It integrates 3 kinds of information. It receives information from the somatosensory information from the spinal cord, it receives motor information from the cortex, and it also receives balance information from the vestibular organs of the inner ear. It has diverse functions. So, for example, it integrates timing and patterning of skeletal muscle contractions during movement, and it is involved in prediction and timing. It controls your posture, maintenance of posture. It also coordinates head and eye movements. It is involved in fast movements or open loop movements called ballistic movements. So, because a lot of our movements are closed loop movements. So, for example, if I want to hold a cup and I want to move my hand to hold a cup, I am looking at the position of the hand at every instant, at every point in this movement, and I am looking at the position of the hand with respect to the cup, and with that feedback, right, I adjust my hand position. But for certain kinds of movements, you do not have the time to take a feedback. For example, if you are jogging or sprinting, right, your steps are, you are laying your steps so fast that you do not have the time to wait for the feedback that comes from the footfall. Okay, so, such movements are called ballistic movements, and cerebellum is thought to be involved in ballistic movements, and so it is necessary for making these fast movements. So, this schematic shows one aspect of cerebellum function. but it does a lot more than what is shown in the schematic. So, for example, when you are moving, right, your motor cortex sends a command, the motor command to the spinal cord, and then spinal cord then sends the commands to the muscles which produce the movement. So, at the same time motor cortex also sends a copy of the motor command to the cerebellum. It is called the efferent copy, right, of the motor command, which is sent to the cerebellum. Then cerebellum takes this command and is thought to implement what is called an internal model or a forward model. So, basically it takes this command, and inside cerebellum there is machinery which takes this command and estimates what will be the expected position of the hand or legs or whatever end effectors which are being controlled by this motor command. So, the spinal cord gets sensory input about the muscle position, the muscle lengths, and basically the postural information or proprioceptive information from the body, and sends it back to cerebellum. Then cerebellum receives this motor command, the copy of the motor command, and using this internal model it estimates what is the expected position of the body. So, cerebellum compares the expected position and the actual position which comes from the spinal cord. It compares these two, and if there is a mismatch between these two, then the motor command did not succeed in producing the correct configuration of the body which it is seeking to produce. So, therefore, this error between the expected position and the actual position, right, is sent back to the motor cortex, right. Therefore, the motor cortex adjusts its command and maybe gives a stronger input or a weaker input, so that to the hand or the legs, so that the expected position matches the actual position. So, damage to cerebellum has associated with can be seen in lots of symptoms. So, for example, since cerebellum is necessary for making fast movements, right, movements which involve fast movements, right, the patients will find difficulty in making such movements. So, one such clinical manifestation of this inability is called dysdiadochokinesia, right, which refers to making fast alternating movements, something like this, which is a kind of movement that you see in when Carnatic musicians, right, sing. Then there is also dysarthria, which refers to impaired speech production. So, cerebellar lesions also give rise to intention tremor. So, it is a kind of a shaking which gets intensified when the person reaches the intended goal position. So, you suppose you are trying to reach some object, right, and there is a cerebellar lesion, right, and the person is suffering from intention tremor. Just when the hand is approaching the object, then the tremor intensifies and hands start shaking intensely. There is also a problem with eye movement, or what is called nystagmus. So, cerebellar nystagmus, which refers to the tremor of eyeballs, right, and this again is intensified. This kind of tremor is intensified when the person tries to make fixate on a given point on the scene. Then there is medulla, which is located rostral to the spinal cord, and we talked about medulla and pons in the last lecture, right, and it is between the spinal cord and the cerebrum, right. There are a lot of centers in medulla which control cardiac function and which these are called vasomotor centers, and it also participates in regulating blood pressure and respiration. So, pons is, you can see in this picture, pons is here and medulla is here, and pons lies rostral to the medulla, that is upwards to the medulla, and it has lots of neurons that relay information from the cortex to the cerebellum. So, these huge bundles of fibers called the peduncles, right, carry information from the cortex to the cerebellum. So far we talked about lots of different components in the brain. So, we talked about sensory areas and motor areas in the last segment. In this segment we talked about several subcortical structures, but you must remember that this kind of a bucket list kind of approach to brain structures is not a very convenient way of thinking about brain structures. We should quickly refer to them as parts of systems, right. Then it seems more logical. So, you can have more logical understanding of the function of these components. So, for example, now if you are talking about the entire motor system of the brain, then at the highest levels of this motor system you have the premotor areas, the supplementary motor cortical area, and so on. and these then send projections down to the motor cortex or the primary motor cortex, which in turn sends projections to the brain stem, and then which in turn sends projections to spinal cord, and spinal cord is called the final common pathway, right, through which all motor output should pass. So, then so when spinal cord neurons, the motor neurons of spinal cord send directly projections to the muscles, and which when activated produce movement. So, in addition, on the sides you have two loops. So, from premotor or the supplementary motor area you have projections to the basal ganglia, and basal ganglia in turn projects back to both the premotor and higher these higher motor cortical areas, and also to the primary motor cortex. On the other side, similarly, you have cerebellum, which receives input from these higher motor cortical areas and then sends feedback, right, just like basal ganglia, back to the same motor cortical areas. So, this whole network of areas form the motor system of the brain. So, similarly, we looked at the visual system. Here also you have a huge network. So, you have the input from the eyes, which goes to LGN, which is a part of the thalamus. So, from there LGN projects to V1, and then from there V1 projects to V2, and then you have the Ware pathway or the dorsal pathway, and the Watt pathway or the ventral pathway. So, there are actually lot more areas than this. For example, the primate visual system alone has something like 120 visual cortical areas. So, it is a fairly complex system, right. So, you can think of mini such systems, but whatever system that we have discussed in these two segments of this lecture, I have just tried to put them all together in a simple cartoon picture. I mean, real brain is a lot more complicated than this, obviously, but I thought let us put all these structures in a logical framework, right. So, in this picture you can see that there are three sensory hierarchies: the touch, the sound, and the vision, right. So, here again you see the primary first, and then secondary, and so on and so forth. So, here neurons in the lower layers respond to more primitive features, and as you go higher and higher in the hierarchy, you find neurons which respond to more and more complex features of that sensory modality. At some point the sensory modality starts blending, starts mixing up, right, mixing with each other, and here you have sensory association areas or this kind of mixed areas. As you go higher up, then in the parietal areas, in the posterior parietal area, we have seen in the last lecture that on the left side, in most people, right, you have a language understanding areas, that is basically you are extracting language related information from these three sensory modalities. On the right side, in most people, right, you also extract large scale spatial information, which is what is processed in the posterior parietal. Now, these abstract information which is extracted from sensory information then is sent to the prefrontal areas. This is where decision making is done. We thought of prefrontal area as some kind of a gate which decides what to do with the information that comes from the sensory cortex, and then based on the decision it sends out motor commands which go through multiple motor pathways. So, you have the skeletal motor pathway, which is what we normally call the motor pathway, right, which goes to the supplementary motor area, and then through to the various motor cortical areas, and then it goes down to brainstem or spinal cord and produces movements of extremities and of the axial muscles and things like that. Then the other pathway is related to speech output, and this activates a Broca's area and produces speech. The third pathway in this picture refers to the movement of eyes, right, or saccadic movements. So, here the cortical areas are like the frontal eye fields or supplementary eye fields and other higher motor cortical areas which are responsible for controlling eye movements. So, these are mainly the sensory and motor cortical pathways. In addition, we have also talked about three loops, right. So, we talked about the loop from the cortex to hippocampus and back to the cortex. We talked about the loop from the cortex to the basal ganglia and back to the cortex. We also talked about the loop from cortex to cerebellum and back to the cortex. The only thing is cerebellum also receives feedback from the spinal cord, which is not shown in this picture. So, like I said, this is a very simplified cartoon picture just to put everything that we have learned in the last two segments of this lecture, right, in a kind of a single framework, right. So, let us summarize some of these main organizational principles of the neural systems that you find in the brain. 5 principles have been described typically, right. These principles are as follows. The first principle is that each functional system in the brain involves several brain regions that carry out different types of information processing. For example, if you take visual system, there is V1 which does primary analysis of the image that it receives from the eyes, then MT performs motion analysis, IT does object recognition, and so on and so forth. Then principle 2 says that there are identifiable pathways that link the components of each functional system. So, in the previous principle basically says there are distinct modules where each module does something specific. The second principle says there are specific pathways which connect these modules in some kind of a logical order, and the processing happens in that order. So, for example, the what and where pathways are examples of these kinds of identifiable pathways that link the various components. The third principle says that each part of the brain, each of these modules projects to other modules in a logical and orderly fashion, thereby creating topographical maps. So, we have talked about maps, right, brain maps in some of the earlier lectures, and next lecture we will talk about them in greater detail, right. This kind of orderly projection from one component to another through what we call point to point connections, right, preserves the spatial information, right, of the representation in one module and takes it forward to the next module. Then the fourth principle basically says that there is intrinsic hierarchy, right, in these functional systems. Like we discussed earlier, if you look at visual system, the neurons of V1 respond to only lines or edges. If you go to V2, you find neurons which respond to curves, right, or angles where multiple lines meet. And if you go further to inferior temporal area, you will find neurons which respond to complex objects. Similarly, in the motor side, as you see in this slide, if you activate neurons of the supplementary motor area, it produces a complex sequential movement, like for example waving of hand or something like that. Or if you stimulate neurons of the premotor area, it activates like maybe multiple muscles or a whole joint. Or if you come down further, if you activate neurons of the primary motor area, it might activate only single muscles. So, there is a hierarchy, right, in the control in the motor system. The fifth principle says that functional systems on one side of the brain control the other side of the body. So, there is a very peculiar pattern in which the functions are mapped onto the brain. And so, if you look at the visual system, the left part of the visual field is mapped onto the right brain, on the right occipital lobe. So, similarly, the left part of the body is controlled by the right motor cortical areas. So, there is a very interesting contralateral mapping of body and the world onto the brain surface. So, information from, for example, information from the left visual field goes to the right hemisphere, and right part of the body is moved by motor areas in the left hemisphere. So, in summary, in this segment we talked about some of the key subcortical systems like thalamus, hypothalamus, basal ganglia and hippocampus. We also talked about cerebellum, although it is not a subcortical structure, right. And we also tried to put all the brain components that we have discussed in this lecture into one grand framework, and we also mentioned that it is an oversimplified picture and real brain is a lot more complicated. So, in the next lecture we will talk about the brain maps and some of the mechanisms by which these maps form. Thank you.